Hey guys, so my name's Lauren, and today we are talking about DNA structure and DNA replication.
So in DNA structure, there are three main types of ways. And so the first one we're going to be talking about is double helix. And so a double helix looks like this. And so basically you have your nitrogen bases, and it's adenine, thymine, guanine, and cytosine. And so they're all within like the DNA structure, and they're all in base pairs. So like AT and GC go together in each section. Okay, so the next one that we're going to be talking about is the phosphodiester bond. And that just looks like this. So they're like little hexagons. And basically they're just all the sugar molecules that bond together within the DNA. So like you have your thymine here, your adenine here, your cytosine here, and then your guanine here. And so these are like the oxygen, and there's like the sugar molecules that just bond together. Alright, so the last one for DNA structure is the major and the minor grooves. And so they look like this. And these are just the bonding sites right in here and right in here. And they're all throughout the structure that bind the proteins during the transcription process, which is the copy of RNA to DNA. Okay, so DNA will store your gene information that is brought down from your parent cells to the daughter cells, and basically it's just being copied over by those DNA molecules. So each person will usually have 46 chromosomes. Not everyone does, but that's what like the regular thing is for humans. So they each come in matched sets of 20. Okay, so there are major enzymes in DNA replication, and they are found within the eukaryotic cells, which are going to be required to use a primer to start the synthesis. And so those primers for RNA get placed with DNA nucleotides. And the primer is going to be used for a template to start the end of the sequence in DNA replication. In DNA replication, there's proofreading. And proofreading is just when the DNA strand can check itself for any errors in the strand. And if there's an incorrect base, there can that's what can like cause those diseases that can get passed from like family history because there was an incorrect base, so the strand won't be right. Sometimes there's going to be like a mismatched pair, and so those will repair the proteins that were in the base and are going to remove it so that the strand can become correct. So in DNA, there are DNA mutations, and those are the change that will occur in the sequence. It can be either from a mistake, or like a mistake when the DNA is being copied, or just like something like a person smoking cigarettes. DNA mutations can be inherited from like genes, and they can be passed down through family members. So it can cause like diseases like cancer or something, and it can affect how the person lives. So there are major types of mutations while the molecules are adding the nucleotides. And so basically there's insertion, translocation, deletion, and then substitution. So basically they can occur whenever there's like an error in the strand. Okay, so basically that is DNA structure and replication. Basically we learned about mutations, primers, double helix, minor and major grooves, genes, proofreading, mismatches, all the stuff in DNA strands that will tie into DNA replication and structure. So I hope you guys enjoyed this video. Thanks for watching.